Okay, welcome everybody to another one of our KBase webinars. If this is your first webinar, then welcome, and we hope it won't be your last. We've been doing these every month now for the past year or so, and it's been nice to see some of our regular users as well as new users come in to learn about the system. So my name is Ben Allen, and I am the outreach coordinator for KBase based out of Oak Ridge National Lab. Miriam Land, who is part of the bioinformatics group at Oak Ridge National Lab and also part of the KBase team. And you probably have interacted with Miriam if you've ever used our help desk. She's here as well, and so she'll be talking a little bit later. But yeah, we're happy to do this webinar today because you know, a lot of these webinars so far have focused on some of the different workflows with KBase in terms of you know, metabolic modeling or metagenomics, and how to string together analytical workflows in the system. And today's webinar is going to be a little bit about that, but what we really want to emphasize are some of the unique features of KBase in terms of where you go after you've done your analysis in the system and you're trying to share that data with a broader scientific community, and in particular focusing on genome announcements, because this is, I'm the person that watches all of the publications that come out from KBase, and this is just something that we see more and more people using the system for, is to prepare genome announcements for isolate genomes. And we've started to see some sophisticated user behavior that we want to highlight and encourage others to emulate, as well as show you some of the ways to do that in terms of sharing your narratives, sharing your data, and really promoting your own use of the system to others. Just quickly, what you're looking at here is our questions and answers document. We'll post a link into the chat. And this has a couple different things. First of all, it has links to the content that we're covering today. So the narratives that we're going to look at, the blog post that has you know, a real good summation of everything that we're going to talk about today, as well as protocol papers that we've written to help you through this process. We encourage you to use this document to ask questions so we can provide answers to them. So everyone should have edit access. So if you have a question, rather than use the Zoom chat, please do not use the Zoom chat. Write it in this Q&A document. And that way we can capture our responses, your questions, and improve our documentation after the webinar is over. So with that, I'm going to go ahead and talk a little bit about this blog that we had written here. So we sent a link out to this a few months back, I think, and this is something that we're trying to do more as a project, is to highlight some of the different aspects and unique features of the systems in a sort of written form. And spoiler alert, we have a brand new website that's going to be coming online in the next couple of weeks, maybe even sooner, where we think it's going to improve access to our documentation and to our, you know, helping you understand the system and how to engage with us and collaborate with us a little bit better. And this is sort of a prototype that we had put together for that, that is on our current website. But the idea here is to tell you a little bit about the way that KBase can be used for creating genome announcements and publishing them. So everything that we're going to talk about today is in here. I'm not going to like read this out to you all, but just wanted to highlight it. And you can see we've got some, dynamic, GIFs and things like that to show you exactly what I'm about to show you. So I am going to start and talk to you all about just basically the upload procedure in KBase. This is something that a lot of users get stuck on, and it can be a little bit tricky. But I wanted to show you all how to upload things just end to end here, starting at square one, and then talk a little bit about our tools for QC and assembly and annotation and things like that. So I'm going to open up a new narrative here, and actually, rather than use my tricky way here, I will open the way that you all probably tend to do it, which is from your dashboard. So once this loads here, I'm going to make a new narrative using this new narrative button. There we go. It's being a little fussy and slow right now. Okay, so I have a brand new narrative here, and what I want to know is how to get my data into this narrative. I've got it on a hard drive or somewhere, or today I downloaded some example data from the JGI actually, and just wanted to download a isolate draft genome that I could use to demonstrate today. So thanks again to the JGI for always being one of our great DOE partners here in systems biology. So I have downloaded onto my computer here some data from JGI. You can see I have some raw data. This is just raw sequencing reads, some QC filtered raw data, and a genome assembly. So how do I get those into KBase? Well, I see I'm in the narrative here and I have a add data button. So I'm going to start out by clicking that. And then I'm going to go over to this import tab here. And spoiler alert, again, I've already uploaded this data into KBase, but I just want to, again, show you how to do this quickly. So I'll start with a small object. What I've done here is actually dragged and dropped this whole folder, and you can drag and drop whole folders. I mean, if you have your own sort of file system with how you stored your genomes, you can drag that into here and it will replicate your file structure. But we're just dealing with a small piece of data here. So I'm just going to drag this assembly here. And we can see it's uploading very quickly and it completed. That's a small object, and so it went very fast. If it was a larger object, it would have not gone that fast. And just want to warn you, and there's a warning here, somewhere on here about this, but you do not want to close or refresh your narrative when you have dragged and dropped data and it's uploading here. If you do that, it will break the upload process, and it will still show the data, but it will be smaller. The size here will be smaller than whatever it is that you intended to upload. So just as a note, if you're dragging and dropping and you're waiting for something to upload, please wait till it's done before you close your browser, close your computer, change your network, anything like that, because you just don't want to break the connection to the narrative. So I have my data here, and I want to import it into this narrative. So I can see a couple different things here. One, I can hit this drop down and I can do things like examine the first 10 lines or last 10 lines of this just to make sure that it's a complete file. But really, I just want to get it into my narrative here. So I'm going to go over to this drop down menu. And I'm going to select what this is. So this is an assembly. You know, if we were doing reads, this would take a little bit longer. So that's the only reason I'm showing you with an assembly. But generally, the process is the same for anything. And you can see all the different types of the data types that you can import here. So once I've selected the data type, I am going to click, I see these 3 buttons here. This one is for importing into the narrative. This one is for downloading out of the staging area, as we call this. So I don't know if I said this. This area is what we call the staging area. And you can think of the staging area as an intermediary between the data you upload and any of your narratives. So you can import things into your staging area and then drag them into different narratives more easily. But for this case, I'm just got one piece of data and I want to get it into this narrative. So I'm clicking the import button there. And what it does is it opens up a cell in my narrative where I can set some parameters for this piece of data before I bring it in. So, you know, I can set some metadata about what kind of assembly I'm bringing in. So in this case, it's a draft isolate. I can set a minimum contig length. In this case, you know, this has been filtered and processed, so this likely doesn't matter very much. But if I wanted to set a minimum contig length, I could do that as well. And finally, I want to set a name for it. So I'm just going to give this one the name example assembly because I have this data importing elsewhere at the moment. But then I click the run button. And what you can see is that it is starting this run up. And right now it's in queue. So depending on how many other people are in front of me in the line, this will take different times. Usually it doesn't take very long for this kind of file because it's relatively small. And we have separate queues for separate types of files. So. But that is going, and while that is going, I am going to actually go to the other narrative that I have here, where I have already done this. So as you can see here, I have imported this this assembly here, and I've got a proper name for it and all of that. In the configuration, I gave it a proper name and all of that as well. And so it's imported into my data panel here. I have the ability to look at information about it, rename it, delete it, download it, etc. And in this, so what I had intended for was to show you all an end-to-end process on this example data, but it's still importing here. So what I'm going to do now is jump over to another narrative quickly, where I just want to show you how the sort of point-and-click operations for some of the common processes that you're going to want to run if you are putting together a genome announcement. So in this narrative, I've got some paired in libraries. So if you, right, for most of you, this is going to be where you start. Maybe you have some sequencing done at JGI or your own university sequencing facility, and they have given you access to FTP or some site where you've got your reads, your assemblies, and other things. So starting at the level of reads in KBase, we can really go all the way through from reads to having completely assembled and annotated genomes. So what I've got here is a reads file let's say I want to only see the types of tools that I can use to process a reads file. So I see in the bottom left-hand corner here, the apps panel, where I've got all of these different categories of apps. And, you know, let's go to read processing, for instance. I can just see there's a ton of different tools here. But maybe I want to kind of filter this whole list down to just things that can operate on reads. So I'm going to click the white space, or you can also click this 3 dots here. And I see this very first button that says show apps with this as input. So I'm going to click that. And what you can see is it put some search text into the search field. That's just an operator that filters the list down to things that can take a paired and library as input. So in this case, I have some reads. Let's say I want to inspect the quality of these reads and see if I need to do any trimming. So the typical tool that we're going to use for this is FastQC. So I am going to click the title of the app here. And I am going to go to the input section here. And what I can see is I have 2 sets of reads. I have the reads and then a trimmed set of reads. So I've already done some of the processing here, but just to show you the example. So I can select the set of reads that I want to assess with FastQC, and then I can click run. And it's sending, and launching, waiting in queue. So I've got another instance where I've already done this here. So and like just for reference, I'm going to be hopping between these kind of demo narratives and this narrative a lot, this genome analysis tools and features narrative. This narrative, which so I've got it here. This is a narrative that I have made to try to capture everything you can do at the level of genome analysis in KBase. I've done other webinars on this one, so I'm not going to talk too much about the content here, but I just want to draw the attention to some of the content in number 2 here in particular, this like drafting isolate genomes, quality assessment, trimming, assembly, annotation. In this section, I've got, as part of this larger example, instances of all of these apps that I'm showing in the other narrative. But so as you're following along, you may just want to keep this narrative open so you can point and click and explore on your own. But anyway, so in this example, here's where I've already run FastQC. And what you can see here is that when it runs successfully, it produces a nice little report about the reads. In this case, this is actually the same set of data as I'm using in this other narrative. So you can see that, well, first of all, you can see I'm a little cramped in this inside this narrative window. So I'm going to go view report in separate window, and voila, it opens up a separate window with the full view of the report. So this is showing me something about the quality of my reads. In particular, I'm looking at the Fred scores across the position in the aggregate of all of these reads. And I can just see that, and this is fairly typical for Illumina data, that toward the ends, the quality is falling off. So this indicates that I probably need to do some tripping on the tail, clipping on the tail end to bring these values into the green zone here, the high quality reads. Otherwise, there's some guides. Again, I've got links to guides on how to interpret all of this in the demo narrative. But what you can just see is that this gives you some sense about how you need to process your reads. So going back to our demo here of just the pointing and clicking aspects, let's say I want to do some trimming. Well, I can see I've got a lot of different processing tools here. The most common tool that we would use for this is Trim-O-Matic. So I can set, again, I open up the app by clicking the name. It loads the cell into here. I can select the data that I want. You can see there are lots of different adapter clipping options. You know, I can automatically remove adapters from these Illumina libraries, set different thresholds, etc. We try to set everything or set default values that are generally good, but right, this is you know, nothing is, every set of data is different. So you know, having some sense of how you want to set these values is important, and being able to interpret this FastQC report is really essential for doing that accurately. So again, I already have this data. So let's just say I set these values and I ran it. And that's where I get this other set of reads here, this trimmed set here. And I want to do some assembly on it. So again, I still have this filtered to show me only things that can take the paired in library as input. Let's go to genome assembly, and I can see, wow, we've got quite a few assemblers here. For isolate assemblies, the ones here that you're going to want to care about the most are spades. You know, Velvet is at this point a kind of a legacy one. You can also use HitMer for assembling isolates, but I honestly, for microbial data, don't necessarily recommend that. Just with this current version that we have, I think it works great on fungal data, and it works great on eukaryotic data and metagenomes, but it can be a little overkill, I think, for just isolate reads. So really, spades, Velvet, and if you have hybrid spades too, if you have a combination of long and short reads, you can also do a combined assembly with hybrid spades. So, but for most people, again, if they just have short reads from Illumina platform, which is very common, assembling with spades is probably going to be your best bet. So after you've done the trimming, you want to put those trimmed reads into spades. Again, we set some standard, some default parameters that, like for instance, it will do error correction. It will do automatic Kamer size selection depending on the size of the reads. But if you want to set custom Kamer values, you can. You know, I just would recommend checking out the Spades manual to do that. And let's just say that you're here and you're like, where's the Spades manual? You can always check out more info about any of these apps, and there's a couple ways to do this. One is you can click this three-dot button here and go to info, which will show you some information about this assembler, link you out to any documentation. So this will take me to the Spades website where I could, you know, find the manual and read more about their Kamer size selection parameters and things like that. But so again, I feel pretty satisfied with this. So I'm just going to say example spades .contigs and click run. And then I'll fly back over to our already done example here where I've got some information about the assemblers and so on. But what you can see here is when it completely assembles and gets where we want it to be, it produces a report here with this tool Quast. You can see information about the number of contigs, N50, L50 values, etc., to get a sense of the quality of your assembly. Here I just want to do a quick plug for a protocol paper that we have written all about, and this is a little dated because I think it's from 2015 or 16, but overall you'll find some useful information about assembling and annotating genomes and some of the strategies around doing that. So again, it's in the Q&A document here if you want to take a look at it. And again, there's information about some of this in inside of these narratives. And that, yeah, there's a, oh, well, I didn't complete loading it here, but there's a corresponding narrative tutorial with information about assembling and annotating prokaryotes and how to read the FASTQC report, etc. And you can find all of those in the dashboard under the tutorial narrative section. After you've assembled the genome, the next step is annotation. And this is the last part of my little spiel here. So actually going back to this this other narrative here, I want to just show you the two annotation tools that we have for isolates here. There is annotate assembly and reannotate genomes with Proca, and this one called Annotate Microbial Assembly. Fortunately, the name of this one is changing soon to be a little more clear to Annotate Microbial Assembly with RAST, because as you can see in the subtitle here, this is built around RAST. So after I have some assembly here, you know, after the spades or whatever has completed its job, I can set this up to do annotation. This is a different organism here, so I want to give it, this scientific name section, one of the reasons you should always fill it out is there are some of the tools for, that we're not talking about today, but for things like phylogenomics, that you're definitely going to want to put something real and not just type in ASDF or something in there. And again, with these tools, we set the parameters by default so that they will run to give you something. If you click the show advanced parameters button, you can set more specific values. For instance, if you want to set genus-specific databases with Proca, or you know, restrict the annotation of certain RNAs, you can do that. And this one overall looks ready to go, so I click run, and it starts to launch. Similarly with RAST, you know, I want to set the scientific name here, and I can set advanced parameters if I want to. In this case, it's more about filtering out which RNAs or CRISPRs, et cetera, that you don't want to call forward in the annotation process. So again, let's just say I've run those. What do I get out of them? I get annotated genomes, which are very nice. here I get, first of all, I get some report about what was done. And this is where it starts to get into some of the things that you're really going to want to include in your genome announcement. And Miriam's going to talk about this just a few minutes, but these reports give you some pretty rich information about which features were annotated, how many hypothetical proteins versus proteins that it was able to pull a functional annotation to, and you can of course compare the two reports between these two apps here, right? This is the RAST-based annotation tool, and here is Proca. Similar, they produce similar reports with a little bit different lingo, but again, you're mostly looking at how many genes did it predict, how many of those that it predicted was it able to assign annotations to, which ontological terms did it pull from for those assignments, and things like that. So as far as, let me just click this here, and you can see it opens up the actual genome data object for this annotated genome. So right, I've kind of gone through showing some of the point and click stuff and showing you some of the reports that you can expect to look at. But really, this is what you're going for, is to have a genome that you can look at the features, look at the content of that, for plugging into different tools inside of KBase, whether that's metabolic modeling or comparative genomic analysis with some reference genomes. At this point, you have something that you can start to explore in that way. Some common things that you'll plug it into right after you've produced the genome are check-in. This is, of course, a very popular tool for looking at the completeness or contamination of genomes. So you can see this is pretty good. And we're looking to build some more advanced tools for exploring genomes. Right now, we have a lot of apps that allow you to process this data and generate some annotations or layers of data on top of that or from the content of your genomes. But we have just recently put up this circular genome visualization tool where you can customize some of the content for printing a graphic representation of your circularized genome. Okay, I've shown you a lot, and I know that's probably like drinking from a fire hose, but just to recap, what we showed is a little bit about the upload process, how to select apps, how to run apps, how to take the outputs of apps and put them into inputs of other ones. And overall have kind of shown you some of the common tools for that you'll use in a genome announcement workflow. I'm gonna pass it to Miriam for the next half hour, and she's gonna talk a little bit about some of the statistics and strategies for in a little more detail for this workflow that I went through pretty quickly here. And then I'm going to come back and talk a little bit about publishing these tools. So I'm going to stop sharing my screen. And Miriam, are you there?
I am here.
Great. Feel free to share your screen and take it away.
Okay. So once upon a time, I was only part-time on KBase, and I was also a user of KBase. And the first thing I'm going to show you came a result of me using my non-KBase time to submit an app to KBase. They allow third-party developers to add their own. As a user, I could see over here on the genomes that your download options were GenBank. At the time, they weren't GFF, and you couldn't send it to staging. This is, the staging option is in here when you have a really massively large genome. It works better if you first put it into staging. That wasn't going to help me with my coding tasks. And when I look at the list of features, there are days when I want to be able to see the whole list and not just put in some keyword like this helicase. There were other things I wanted to be able to do. As long as I'm here, I'm going to point out that if you click on these IDs over here, it'll open up a new tab to show you more about that gene or CDS. And I want to point out the protein translation is here. I copied this and I'm going to use it at the very bottom of this narrative. I'm going to come back to this later. So one of the things I wanted to be able to do was get a list of all of the genes. And I bring this up because this particular app is going to be going through a name change shortly. It's going to become officially part of KBase instead of me as a third-party developer. And these names are going to change. And the thing that is going to be consistent is the word report, if you want to search on that. And in this case, you pick some genome. I happen to have 2 in this narrative. You pick either one. And the options are to be able to get the list of features or genes, if that's how you think of your features, as tab delimited or comma delimited. You can get a FASTA of them or a FASTA of the mRNAs. This GFF format was in there. It's left over because it wasn't available at the time I wrote this. And you can also get a FASTA of the DNA, which if it's your own genome, you probably don't need that, but you know, there might be cases somebody asked for that. And the pretty version of the output is what you get with these tables. There. The output from my text reports aren't necessarily pretty. I'm going to show you an example in just a bit. But they are designed to be just plain text so you can download it and use it with other tools on your local computer. They may look ugly on the screen, but they serve the purpose of a tab delimited, comma delimited file. So in this app narrative, I also put together a set of genomes, a genome set of the 2 mycoplasma that I have, and I ran a text report on the genome set. So if you are doing an announcement, you frequently want to be able to compare yourself to somebody else that's published. Like you have more genes or you have smaller number of contigs or something or other. And as promised, it looks ugly on the screen. But if you download this tab delimited file, you can pull it into Excel and get something that looks like this. So you can have one of these nice little tables that you can put in your genome announcement. You could have 30 genomes in here and compare how they are in GC content, number of genes, and blah, blah, blah. Whatever you have put into your narrative, you can include in this table. And didn't realize Ben was also going to include the circular genome visualization. So I am just going to close that. Another way people want to be able to compare their genome to the rest of the world is with 16S. KBase does not do 16S. In many cases, 16S does not have enough resolution because it is one gene comparing across all genomes. And if you're dealing with closely related strains, it just doesn't give you the detail that you need. So one way we have addressed this is this app called Insert Genome into Species Tree. There is a pre-built tree of not every genome in the world, but I think it's around 10,000 genomes. And what it's doing is taking everything you've selected and putting it into the tree so that you have some context. If you have 3 or 4 genomes that you really, really want included in your tree, make sure you include them as part of your input. I'm going to click on that tab in just a second. As a user, One of the first things I noticed is that part of the reference database had a duplicate of mine, and they both come out here, and my two input genomes are sticking out in yellow there. I know there are other people who have highlighted that as an issue, and I think somebody is working on it. But the good news is there are various download formats so that you can download it and put it into your own software and tweak this up to make the tree look like what you want. I'm just going to show you how I configured this. I put in my two genomes. I said I want a neighborhood around them of 20 genomes. And I said, I left this checkbox off. If you check that, all 20 of those genomes that got added, then get added to your data panel. Maybe that's a plus, maybe that's a minus. I didn't include it in my analysis here. And another way that is quickly becoming popular for comparing a set of genomes and closely related genomes is to use an ANI process. So we have fast ANI. And again, you just configure the genomes you want included in that. So you could have included that whole list of 20 neighborhood genomes if you had added them to your data panel. And these two PDF links here look something like that. I'll let you decide whether or not that is something you want to use in your report. Another thing you might want to do is compare proteomes. And while this sounds like a great idea, I'll tell you, this is all you get right here is this nice little old-fashioned dot plot. This was designed to primarily be a way to navigate through here and do online. It is not for taking a nice little picture and downloading it and slapping it into your genome announcement. And it is strictly a two-genome process here. I don't want you to spend time on this and then be disappointed. It has its place, and I'm not going to try and navigate through here because it's easy to get lost if you're trying to do this live in front of other people. But it's primarily for doing online work, not for publication. What we have instead is the ability to compute pangenome. Put in your little genome set and you get this little table here of output that compares the homologs and the singletons that you have in your set of genomes and how they compare here and the families of genes. And there we go. Unfortunately, we just learned recently that if you click on this, these two sorts over here, you're going to get an error. Something on the back end changed, so now the UI is producing an error. So the first thing we're going to do is remove these sort buttons, and then we're going to fix the problem. So eventually that will be fixed. But you always have the option of downloading the pangenome if you'd like to do that. And a pangenome can be downloaded as Excel or a tab delimited file if you want to be able to sort this in some other way. The other thing that we have discovered about pangenome is somebody recently tried to do 332 genomes in their pangenome. And it calculated the pangenome fine, and they'll be able to download it. But when they got to this table here, this is where they got the red error. It's a new problem. We're working on it. What we always have people testing the limits of what KBase can do, and eventually we discovered that there is an upper limit, and we work around it. You can also build a pangenome with orthomcl. If you're familiar with orthomcl, it has its own format output, and it doesn't have this nice little table output here. For people familiar with it, you can download in what orthomcl gives you. So the person who put in this particular app chose not to use the the table here as part of the output from this pangenome. But what you can do is run an app called Compare Genomes from a Pangenome. So it will accept input on either of those two types of pangenomes. And the output has one more column in it. It has these functions. I haven't yet tried whether or not all of these sorts here work. Since I just found out about the sort problem yesterday, I haven't been brave enough to try that yet. If they aren't working, we will have to put that on the list of things to work on. One of the things you can do with a pangenome is to do a circle plot here. Obviously, 332 genomes are not going to work in this amount of space. I can't even imagine how many pixels you would get out of this, but I think you could comfortably do at least 20 to 30 in a circle plot. I don't think I have tried more than 20 myself. We also have ways of doing domain annotation. So the output itself isn't very elaborate there, but you get a domain annotation that you can download if you want, comma delimited format. And under the options for annotating, I forgot. There is annotate domains in a genome set, but there's also annotate just one genome. If you're annotating just one genome, you get more options for only doing Cogs or only doing PFAM or only doing TigerFAM. If you do a genome set, it just automatically does them all. Kind of handy that way. What can you do with the domain annotation? You have this view functional profile. Help if this was at the top for the genomes. So when I configured this, I said I only wanted Cogs. So I'm just getting the Cogs output here. And obviously that right there is, let's see, J. What is J? Translation, ribosomal structure, and biogenesis for my two genomes. Yeah. So in the configure, you can say which of these you want. I would have to reset this and rerun it to have these links be live, but you can select which ones you want in your output. I am going to skip that because I just found that it is not working as described. One of the things you can do is focus in on a set of genes that you want to analyze, a gene family. Usually there is some functional process, or set of enzymes, something that caused you to sequence a particular genome. So I built a feature set for the first genome, and this is me configuring a feature set for the other of the two genomes. And after you select which genome you want, it gives you a drop down of all of the genes that you can choose from. And I picked helicase. And what it's showing me here is that I've already selected all of those. I can remove them if I want. So green are things that I can add, and red are things I can delete. I've already run this and created a feature set, so a set of genes. And I merged the two feature sets from my two genomes into this, and so I have a total of 8 genes, 4 from each of the genomes. And now that I have those 8 genes, one of the things I can do is a muscle alignment, a multi-sequence alignment. And Ben showed you how you can do this to say, show me all the apps where I can use this as input. So among the things that you can use this multi-sequence alignment for is a phylogenetic tree of just those 8 genes. So this is a way of getting a figure for your paper. You pull out the genes that you want included from all of your different genomes and do this alignment. Another way that you can use feature sets is in a BLAST. So in this case, I am using my 8 genes as the target or subject in BLAST terminology. And remember, all the way up at the top, I talked about copying the protein. I want to use this as my query sequence. And I can run the BLAST. And yes. And you may notice that even though I had 8 genes in my subject set, I've only got 2 hits. And yet they were all helicases. Sometimes it's because you need to adjust the parameters. I'm showing all of the advanced parameters here. So you could change the E-value and the bit score and the percent identity. If these are truly closely related genes, then changing these parameters would result in more output, more genes in that result tab. If they're too far distantly related, no matter what you change these things to, you may not get any hits. But that is a question I sometimes get as to why is there only 2 in this list instead of the ones that I was looking for. So I could make those really loosey-goosey and then make decisions about which ones I want. And as with many of the others, there is a way to download. This would be a tab delimited text file that you could download your results into. And I haven't looked in a while, but I think this one actually has all of the results, not just the ones that pass the criteria here. And I think I am ready to pass it back to Ben.
All right. Great. Sorry, I was just entering a question there. Let me share my screen again. All right. So what I just wanted to talk about now is some of the publishing and sharing side of using KBase, and this is something that we think makes our platform different, and I think there's a good case to be made for that I'm going to show you more about in a second. But really, what we want KBase to be is to help increase reproducibility, to enable fair access to data, and accomplish these kind of general goals that a lot of agencies, a lot of research groups are aligning with right now across the genomics field, to help contribute to that, the narrative system that we were showing all of these analyses. And someone asked at the beginning, what is the narrative you're talking about? And this is just to make it clear, every single one of these full analysis with data and apps and content here, this is what we call a narrative. And so in KBase, as you do your analysis, you generate these narratives. And what we want each of these to become or evolve as you are doing your analysis, we want them to evolve from something that's more like a lab notebook into something that's more like a publication that you are trying to share with your peers, with your collaborators, with the community at large, and for each one of your narratives to become a rich resource of data and analysis in and of itself. And so what we're excited about right now is that we're seeing users start to realize this aspect of the system, and they're starting to share their narratives, and genome announcements is one of the ways that we're seeing that happen the most. So I'm someone that watches all of the publications that come out using KBase, and you can, of course, I was just showing the how you get to the publications that use KBase on our current website. And one of the things I'm starting to see more and more is people sharing these narratives as a way to both distribute the data, distribute the process that they use to analyze their data. So sort of giving access to the exact same place in space that the analysis was conducted. So that kind of enables the reproducibility. And yeah, they're using KBase as a sharing platform for their data and analysis. So for example, here is a very recent genome announcement that came out just less than a month ago. And you can see this is, of course, from our colleagues, Mircea Podar and Dale Pelletier at Oak Ridge. They actually did release 2 announcements that featured narratives. And what you can see, they're kind of talking about the announcement itself and the process that they did. And sure enough, at the bottom, you can see they have given access to the narrative. So I'm just going to click this and show you what they did. So here we're taken into what you can see here is a view-only narrative. And they've set this up very nicely where they have some information about the collection and sampling and sequencing methods. They have their import here. So these app cells are closed right now, so I can expand them to view the configuration or to view any of the reports. It takes a little second to load here, but anyway, this gives me access to, you can see this is just giving the world access really to this analysis that they've completed here, all the way from assembly to annotation to quality checking, fast ANI, just all the stuff that we've kind of talked about. And in addition to that, they've drafted a metabolic model for this genome that they're announcing. So they're kind of including all of this stuff in a single space that they can share with other users. And this is of course very exciting, because this is what we would say is like model behavior. This is what we want to see people doing in KBase more and more. And so, okay, let's say I am someone from the outside coming in and I'm like, well, I can see the narrative, but maybe I want to check it or validate it. How do I get access to it? So I can click this this button to re-drag out here, and you can see that I can't add any apps or anything, but I can access their data. So if I want to download any of these, I want to download the assembly or the genomes, I can just click this export download data button here, and then I can download, the GenBank file, GFF, FASTA. I can just load it into my staging area to re-import it somewhere else in KBase. I can even, I could even make a copy of this narrative. If I just wanted to make a whole copy of it for myself, I can do that and change the parameters, rerun tools, add new tools, etc. So similarly, I can view associated report if I wanted to see any particular specs about this data, like for instance, what is the provenance of it? Maybe not very interesting for this this piece of data, but I'm sure if I do that for the metabolic model here. It might show something a little more interesting. And this is just showing, right, the content of this metabolic model if I want to see the reaction lists, compounds, genes, etc. But here you go. This is like a provenance graph where I can see the data referenced by this data, so the kind of foundational data going into it, the model itself, and then anything that's referenced downstream from it. You can see I can just look at information about the tools and the parameters, anything that was used to generate it. So this is just kind of giving you a sense of the real like preserving power of KBase for your analysis when it was done. Because and I say that because right, genomics and computational biology is everything is a moving target. The tools are developing all the time. The reference databases are changing all the time. How those things communicate together is changing all the time. KBase, we have a way of coordinating that, and of course our whole project is built around trying to coordinate those things together in sync. And what you're looking at right here is a snapshot of when and what tools were available for this specific process. So that's important because if you're looking at, you know, even announcements or papers that are a couple of years old at this point, your ability to access some of the data, maybe it's just tucked away on a FTP site somewhere and those links are broken or something, you know, the ability to access the data, you know, it depletes or decays as the technology changes. So everything that you're looking at here is a snapshot of what was done. And there's a way we can actually take an even more advanced snapshot of this that I'll show in a second, but just want to kind of highlight this as again, model behavior. And, you know, there are a couple of other announcements. This is another one from Murcha and Dale, where they put it into the bottom of their microbial resource announcements paper. So anyone can access this. Another one from Dale and Chris Schott's group, a non-Oak Ridge person that did this recently. This is not necessarily a genome announcement, but it's a metagenome analysis paper where this is just a really, you know, rich example of using the ability to craft a narrative with Markdown, with images, with data sharing, etc., and using KBase as a way to share your data and share the analysis that was used to generate the data in a semi-permanent way. So let me go. So the other thing I wanted to highlight quickly, oh, I think I had it in another, okay, never mind. I'm going to go to, so let's just talk about quickly another feature that we have to give access to your data, and that is our organizations. So sorry, it's going slow. Okay, there we go. So from our dashboard on the left-hand side here, we can click orgs, and this takes us to organizations. And organizations in KBase are a way for people with KBase accounts to share, to provide sort of blanket sharing permissions to all of their narratives across a set of users. So I just want to highlight this because oftentimes sharing access to genomes or published data, for instance, out of a whole group is sort of a challenge, right? You may have a set of distributed individual papers, but let's just say you want to kind of give somebody a single place to access all of the isolates and draft assemblies and genomes that you've generated. Organizations are a powerful tool to do this. So I'm just going to go into this top group here for Enigma. It's our PI Adams group, and you can see he's got all this nice stuff about his group here in the about, but I really want to go to get that data. So I click the narratives here, and what I can just see here is where they have started to share access to all of the data that their group has. I'm not part of Enigma, so but I could I can join this organization. I can request to join this organization or any others, and then the administrator Adam will get a notification that I'm requesting access to that data. So I would encourage you all to explore the organizations that we have right now available, and see what kind of data you can get access to on the system from some of the groups that are sharing their data using KBase right now. One last thing. So I've talked about sharing narratives, putting them in your publications and all of that. And I really haven't shown you much about how to do that at the per narrative level. So let's just say this is some narrative that's complete and I want to share it with other people. This is already a public narrative, so let me just show you what that kind of looks like. It's sort of the same in any narrative here, but if I go to the share button at the top right, I can see that there's two buttons: manage sharing and manage static narratives. Well, let's go through each of these, but I'm going to go to manage sharing first. And what I can see is right now that this is a public narrative. By default, any narrative is private. So this is just the one that I was drafting in the demo process. Okay, I'll call it untitled. I know we have a million untitled narratives, but and you can just see it's by default private. So if I want to make it public, I have to click this make public button. And that would convert it to public, just like this other narrative that I have. But if it's public, that means that anybody can come and view the narrative. It doesn't mean that they can edit it or share it. It just means that they can view the narrative, and it does mean that they can access the data. It means that they can download the data. So when you're making a narrative public, your fundamental question should be, do I want to make this data public? Because that's what you're doing in effect, is making the data and your analysis publicly viewable. If I want to share a narrative with a specific user, I can do that. So for instance, let's say Ellen, I want to give, this is a KBase, new KBase staffer. So I want to give Ellen the ability to edit and save a narrative. So I could do that. And now Ellen has the ability to edit and save this narrative. I can, of course, do this, for a public narrative, it's blanket view only privileges for everybody with a KBase account. But if it's a private narrative, you may want to give people specific view only privileges. So I showed you all orgs. Let's just say you want to share your narrative with some existing organizations. You can click the orgs button and you can do that as well. You can go and find some org that you're a part of, and you can share it with them there. And that will give all of the people in that organization access to the narrative. But what one of the, so those are the common ways, and this is what we've seen users sort of take up, like I was showing with those publications. This is what we've seen users starting to do, is use public narratives and linking to those public narratives in their publications to give access to the larger community. A new feature within the past couple months that we want to advertise to you all is the ability to have static narratives. And so, as I've said, even with public narratives, you have to be a KBase user or a KBase account holder to access them. Now, given that we're a free system and it's free to create an account, it's free to do everything that we've shown you all today, you know, there's very low barriers to that. But if you want to even take your narratives even further, we have an exciting new feature called static narratives that really both gives you the ability to share your access to your narrative with people who are not even KBase users. So it creates an HTML web page, if you will, of all of this stuff in your narrative. And you know, we're still working out some of the kinks of this, but it works pretty good for really spreading out your access to your data and your analysis even farther. So I'm just going to click Manage Static Narratives here. It says no static version for this narrative exists yet. Create static version. So I'm going to create a static version of this narrative here, live on the fly. And what it's going to do is give me a link to the HTML version of this narrative that is a snapshot of the whole narrative. Just to give you some more contrast to, okay, well, what's the difference between a static narrative and a public narrative? So if for a public narrative like this one, it is showing not only just the content at any given time, it's showing you the most up-to-date content. So let's say I'm the owner of this narrative and I change something, I add something or delete something. Anyone that has access to this URL, if they come back to it, they will see the changes that I have made, and they will see any modifications that have been made to the narrative. So, you know, if I rerun a tool or I run a new version or, you know, change something up, that means the public will still be able to see that. So static narrative error, that's not what we want to see. Let's see, maybe it did generate it, but okay. Well, that's an interesting problem. So we'll work on that. But let me find another example of this really quickly here. Sorry about that. Oh, I know there's actually should be one in my genome in here. Yes. So I talk about this. Oh no, but I don't actually have a link to it. Well, you can see in this GIF, it's showing a little bit of a preview of that. Let me really quickly find the viral example that.
Hey, Ben, I put one in the Zoom chat.
Excellent, Zach. You are the best. Thank you. Okay, perfect. Yes, Zach put in. Yes. Oh, yeah, that's right. So there's a static narrative for the. This is, I think we're putting this together after the fact, but this was for the one of the genome announcements that I was showing. So as you can see here, anyone can access this, and this is a snapshot of the narrative independent of its current state. So even if they changed anything about the public narrative associated with it, this snapshot remains consistent. So what you can just see here is all of the content of that narrative posted up inside of a way that anyone that has access to this URL can come and see this content. They don't have to have a KBase account. And this is, these are indexable by search engine. So that's the other advantage of putting these out there, is that they are indexable by search engines. So in theory, somebody looking for, just Googling an organism can come across these. And so you can see it's got access to the data here, and they can click it and see more information, or copy it or download it. And it also generates a list of all the citations associated with the apps. So yeah, thanks Zach for putting that in there. And so what we've shown here again is this is a lot, and it's hard to cover a really wide range of tools and features in just about an hour. But again, I just want to refer everyone to this blog, Make the Most Out of Genome Announcements with KBase, where we really try to capture all of this stuff in a more permanent way. And of course, we're going to share this recording of the webinar with everyone afterward. Yeah, I hope this was helpful to you all. And again, would refer you back to this blog post and the other resources that we have put into the top of our Q&A document here. If you are on Twitter, please follow us there. We announce all sorts of stuff on there, whether it's just minor bug fixes, feature updates, training, new publications, etc. We're trying to increase our activity there. And so if you give us a follow, you'll be on the cutting edge of KBase news. So, and finally, I'll send a link to this after the fact, but if you could, if you all would go and fill out this feedback form about the webinar, that would help us improve these as we keep going on. That's all for this one. The only other announcements are that we've got the next two webinars we're going to do. In August, we're going to do a webinar that goes into more in depth about using some of the markdown and publishing features of KBase. And in September, we're going to showcase some of the new viral tools that are coming online to the system. So on the current website, to stay in touch with our webinars, just go to the About page, and we'll post up the next webinar here shortly. Or like I said, even better is to follow us on Twitter. And otherwise, thanks a lot, everybody, for sticking around, and hopefully you found this a valuable event. So thank you.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOEKBase. And if you have questions or encounter an error when using KBase, please contact the help desk.